Hello everyone, welcome to Lab 9. Today we are going to discuss about heart and circulation includes blood vessels and lymphatic vessels. So first let's have some summary questions from last lab. Question number 1, what is this a section of? Be specific. So here we have a section which looks like a dense regular connective tissue. But when we see the nuclei, they are not darkly stained. We can see abundant euchromatin. So this is not a dense regular connective tissue. This is a longitudinal section of smooth muscle. And the muscle is in a relaxed state, so the nuclei are elongated. If it was in a contraction state, the nuclei becomes wiggly or worm-like. Question number 2, name the structure indicated between the arrows. Here also we have a longitudinal section of a muscle and we can see these striations. So this could be either a skeletal muscle or cardiac muscle. And when we see the muscle fibers, they are branching and they contain one centrally located nucleus. So this is a cardiac muscle. And the arrow points into these dark transversely running lines. These dark transversely running lines are intercalated disc. It's where adjacent cardiac muscle cells communicate. So the arrow points into intercalated disc. Question number 3, name the structure located between the brackets. Be specific. Here also we have a very eosinophilic tissue with polygonal cells and multinucleated cells. So this is a transverse section of skeletal muscle, and as you know it, bundles of skeletal muscle fibers or fascicles, they are surrounded by a connective tissue, and this connective tissue is known as perimesia. So the bracket, it points into perimesia, which surrounds these fascicles. Question number 4, name the region indicated by the bracket. Be specific. So this is an electron micrograph of a sarcomere. So this dark line is Z line or dead disc. This one is also Z line or dead disc. So this is one sarcomere. And at the middle of this sarcomere, this relatively dark area, it's formed by both thick and thin myofilaments, and this is the A band. So in today's lab session, our learning objectives are, by the end of this lab session, you'll be able to identify the 3 layers of the heart: the epicardium, myocardium, and endocardium. And we'll also discuss about the Purkinje fibers, which are found in the endocardium. And we'll also see the layers of large and small blood vessels. And we'll also discuss about lymphatic vessels. So let's start with the heart. So histologically, the wall of the heart consists of 3 layers. So the most outer layer is the epicardium, and the middle thickest layer is the myocardium, and the most internal layer is the endocardium. And as you know it from lecture, heart is covered and protected by fibroserous pericardium. So this the most outer layer. This one is the dense irregular connective tissue, and it's a fibrous layer of the pericardium. And the serous layer of the pericardium has 2 layers. We have the parietal layer and we have the visceral layer. And in between these 2 layers, there is a cavity, pericardial cavity. So as you see, the visceral layer of the pericardium, the serous pericardium, it adheres to the wall of the heart and is one component of the epicardium. So epicardium is composed of the visceral layer, which is mesothelium, and connective tissue. So this yellow structure indicates connective tissue. And when you see the middle layer, it's the thickest layer, and it's composed of cardiac myocytes. So that's why it's known as myocardium. Myo stands for muscle. And the most internal layer is the endocardium. So this layer, it faces to the chamber. It comes in direct contact with blood. So it's composed of endothelium plus subendocardial connective tissue. So as you know, heart has 4 chambers. We have right atrium, left atrium, right ventricle, and left ventricle. Generally, when we see the atria, the wall is thinner than the ventricles. But it contains these 3 layers, the endocardium, myocardium, and epicardium. So first, let's see the atria. So this is the right atrium, and this one is the left atrium. You can see the difference in the thickness of the 2 atria. So first, let's see the right atrium. So these bulges are formed by pectinate muscles. In the myocardium, there are pectinate muscles, and these pectinate muscles form these bulges. And these bulges, as you see, they face into the chamber. So we can use these bulges to orient ourselves. So this is the endocardium, and this is the epicardium. So let's zoom in and see the components of endocardium. So this is the endothelium, the inner lining of the heart, and this is the subendocardial connective tissue. So endothelium and subendocardial connective tissue, they constitute endocardium. Then we have a very thick myocardium, which is composed of cardiac myocytes. And externally, we have the epicardium, which consists of mesothelium and connective tissue, underlying connective tissue. So this mesothelium and this connective tissue, they form the epicardium. Similarly, in the right atrium also, we have these 3 layers. So this is the endothelium, and this is the subendocardial connective tissue. And this subendocardial connective tissue, it contains adipocytes. We can also see these adipocytes. And here, this is the myocardium, which is composed of cardiac myocytes, and this is the epicardium. The epicardium is also composed of mesothelium and connective tissue. And in this layer also, we can see adipocytes. And when you see the ventricles, in the ventricles, in the endocardium of ventricles, we have specialized cardiac muscle cells, which are known as Purkinje fibers. As you see it in this picture, this yellow line indicates the Purkinje fibers. They are one component of the conducting system of the heart, and they are found in the endocardium. Normally, endocardium and epicardium, they are more or less identical. So in the ventricles, we can use the Purkinje fibers to distinguish endocardium from epicardium. So the Purkinje fibers, they are cardiac muscle cells, but they are large in size, and their cytoplasm is filled with glycogen. So we can use these 2 features to identify Purkinje fibers, and the Purkinje fibers, they will help us identify the endocardium. So first, let's see a slide with PAS. As you know it, PAS, it stains carbohydrate. It stains carbohydrates magenta color. So this is a ventricle which is stained with PAS. So these darkly stained or these magenta color cells are the Purkinje fibers. So this is the endocardium. So this is the endothelium, and this is the subendocardial connective tissue. And in the subendocardial connective tissue, we can see these Purkinje fibers, specialized cardiac muscle cells. They are different from the typical cardiac muscle cells in that they are relatively larger in size, and their cytoplasm is filled with glycogen. That's why they appear magenta in this staining. And in this section, we can also see one component of the fibrous skeleton of the heart. Here it is. So here we can see a cartilage. So as you know, these are the chondrocytes which lie in lacunae. And this is one component of the fibrous skeleton of the heart. So this is fibrous skeleton of the heart. And this one is the epicardium. So the epicardium is composed of mesothelium and connective tissue. And we have also H&E staining of the ventricles. And in H&E staining, the Purkinje fibers, they appear pale. Normally, as you know, H&E, it doesn't stain glycogen or carbohydrates, so the cells will appear pale. So here, this is the left ventricle, and this one is the right ventricle. You can see the difference in the thickness of left and right ventricle. So left ventricle, it distributes blood throughout our body, so there is functional hypertrophy. So roughly, left ventricle, the wall of left ventricle is 3 times thicker than the right one. So let's zoom in and see first the left ventricle. Even in this lower magnification, when we see the Purkinje fibers, you can see it here, they are pale in staining. But the typical cardiac muscle cells, they are very eosinophilic. But this area is pale in staining. It contains the Purkinje fibers. And the Purkinje fibers are found in the endocardium. So let's zoom in and see. So this is the endocardium, the endothelium, and subendothelial connective tissue, and these are the Purkinje fibers. As you see, they are relatively larger in size, and in H&E staining, they appear pale because they contain a lot of glycogen in their cytoplasm. But when we see this is a typical cardiac muscle cell, so they are very eosinophilic. So this is the endocardium. This very thick layer is the myocardium. And here, most externally, we'll have the epicardium. So this is the epicardium. It contains mesothelium and connective tissue. We can see these adipocytes in the connective tissue layer. Similarly, the right ventricle also has these 3 layers. Here in the right ventricle also we can see this area. It's pale in staining, but this is very eosinophilic. So these are the Purkinje fibers. So this is the endothelium and subconnective tissue, and this is the subendocardial connective tissue that contains Purkinje fibers. So this pale staining, relatively large cells are Purkinje fibers. And this is the myocardium, or the middle layer. And here, most externally, we will have the epicardium, which is composed of mesothelium and connective tissue. Now let us see blood vessels. Large and medium-sized blood vessels, their wall is composed of 3 layers. Most internally, we have tunica intima. And tunica intima is composed of endothelium, basement membrane, subendothelial connective tissue, and internal elastic lamina. The amount and proportion of these layers, it may vary depending on the type of vessel. And the second layer is the tunica media. The tunica media is mainly composed of circularly arranged smooth muscle. And it also contains external elastic lamina. And the content of tunica media, it also varies depending on the type of vessel. For example, if you take the aorta, in the tunica media, in addition to these smooth muscles, we'll find elastic laminae. So the tunica media also varies depending on the type of blood vessel. And the tunica adventitia, it's a connective tissue, so it contains fibroblasts, and these fibroblasts will produce collagen and elastic fibers. And there are also blood vessels and nerves in the tunica adventitia. So the tunica adventitia is the most external layer, so it can't get nutrients from the lumen by simple diffusion. It's relatively far from the lumen, so it needs its own blood supply and innervation. So the blood vessels that supply a blood vessel, they are known as vasa vasorum, and the nerves are known as nervi vasorum. So the vasa vasorum and nervi vasorum, they are found in the tunica adventitia. It's a connective tissue layer. So first, let us see the largest blood vessels, the largest artery, which is aorta, and the largest vein, which is vena cava. So aorta is an example of elastic artery, and vena cava is an example of large vein. So when we see the wall of the aorta, it has a round lumen, a well-defined lumen. Then in the tunica intima, we have endothelium, we have subendothelial connective tissue, some smooth muscle fibers, and we have also the internal elastic lamina. So this constitutes the tunica intima. Then the tunica media of an elastic artery is very thick. It contains 10 or more layers of circularly arranged smooth muscle. So it contains these circularly arranged smooth muscles, 10 or more layers of this. Then in between these smooth muscles, there are elastic laminae. So these yellow structures indicate the elastic laminae. And there is also an external elastic lamina. And the tunica adventitia, it's relatively thin and it's typical. It's a connective tissue. It contains vasa vasorum and nervi vasorum, and it also contains fibroblasts, collagen, and elastic fibers. But when we see the vena cava, the lumen is not regular, it's irregular, and it's collapsed. And the tunica intima, it consists of endothelium, subendothelial connective tissue, some smooth muscle fibers, and irregular elastic fibers. Then when we see the tunica media, it's relatively very thin when we compare it with the elastic artery. The tunica media in large veins is very thin. It consists of only very few circularly arranged smooth muscles. But the tunica adventitia is very thick. It contains bundles of longitudinally arranged smooth muscles, and running in between these longitudinally arranged smooth muscle, we will have the connective tissue. And we can also find the vasa vasorum and the nervi vasorum, the fibers and fibroblasts. So the adventitia is very thin in elastic arteries, but the adventitia is very thick in large veins. So let's see aorta and vena cava in a section. So we have both HND and Verhoef staining. In Verhoef staining, we mainly see the elastic fibers. So as you know, elastic fibers, they are not clearly seen in HND staining. So first, let's see the HND staining. So this is the vena cava and this is the aorta. So first, let's see the aorta. So this is the tunica intima, which is composed of endothelium and subendothelial connective tissue. And here it's not clearly seen because it's an HND staining. Here we have an internal elastic lamina. So from the internal elastic lamina up to here, this is the tunica media. As you see, it's very thick. And this tunica media, it consists of circularly arranged smooth muscles. And in between this smooth muscle, there are elastic laminae. Even with this HND staining, we can see these elastic laminae. We'll see it clearly in Verhoef staining, but even with this one, we can see it. So it contains a very thick tunica media. And this is the tunica adventitia, a connective tissue. So as you see it in the tunica adventitia, we have vasa vasorum, and we can also look for, and these are nerves, so these are nervi vasorum. And now let us see the vena cava. So this is the tunica intima, which is composed of endothelium and subendothelial connective tissue. And this is the tunica media. This very thin layer, relatively thin layer, is the tunica media. But this very thick layer starting from here up to here is the adventitia. And the adventitia, it consists of longitudinally arranged bundles of smooth muscles. These are in parallel with the lumen of the vessel. They are longitudinally arranged. And running in between these longitudinally arranged smooth muscles, this is the connective tissue. And most externally, we will have the connective tissue. And here, this is the peripheral nerve, this is the nervi vasorum, and we can also find blood vessels, vasa vasorum. So we have this same slide with Verhoef staining. Verhoef staining mainly stains elastic fibers. And elastic fibers in Verhoef staining, they appear black. So this is the aorta. So here we have the tunica intima, which consists of endothelium and subendothelial connective tissue. And this black line indicates the internal elastic lamina. And this is the tunica media. So as you see, the tunica media, it contains elastic laminae running in between the smooth muscles. And this is the tunica adventitia. In the tunica adventitia also, we have elastic fibers. And now let's go one size down and let's see muscular artery and collecting vein. So muscular or distributing artery, the tunica intima, it consists of endothelium, very thin subendothelial connective tissue, and internal elastic lamina. Then the tunica media, it consists of circularly arranged smooth muscles. And in between these smooth muscles, in muscular or distributing artery, we can find elastic fibers. They don't form the laminae, but we can find elastic fibers. And then there is external elastic laminae. And the tunica adventitia is typical. It's a connective tissue. It contains vasa vasorum, nervi vasorum, collagen and elastic fibers, and fibroblasts. And when you see the collecting vein, the tunica intima contains endothelium and subendothelial connective tissue with very few smooth muscles. And the tunica media is thin. It contains only few layers of smooth muscle, but the adventitia is very thick. And it contains vasa vasorum, nervi vasorum, collagen fibers, elastic fibers, and fibroblasts. And there are also few bundles of longitudinally arranged smooth muscles, but dominantly it's connective tissue. So an example of muscular artery and collecting vein is popliteal artery and popliteal vein. This artery and vein, they are found in the lower limb, and they are an example of muscular artery and collecting vein. So in addition to all the features mentioned, we can also use the lumen to identify arteries and veins. So the lumen of artery, it's regular and commonly it's round in shape. But the lumen of veins, it's irregular and it's collapsed. So this is the popliteal artery and this is the popliteal vein. Let's zoom in and see the layers. So this muscular or distributing artery, it contains a very thin endothelium and subendothelial connective tissue, and it contains internal elastic lamina. So this squiggly pink line indicates the internal elastic lamina. So this is the internal elastic lamina. And this relatively thick layer is the tunica media. It's mainly composed of smooth muscle, but we can also find elastic fibers in between the smooth muscles. It's not clearly shown here because it's an HND staining. Here we will have another elastic lamina, the external elastic lamina. We'll see it in the Verhoeven staining. And this is the adventitia with the vasa vasorum and with the nervi vasorum. So this is the connective tissue. And when we see this collecting vein or this popliteal vein, so this is the endothelium. This is the endothelium. And under the endothelium, this is the subendothelial connective tissue. And this section, this popliteal vein, is pathological, as it's mentioned in the legend. That's why this subendothelial layer is thick. Normally, the subendothelial layer in a healthy vein is very thin, but this one is pathological. That's why this subendothelial connective tissue is very thick. Then this is the tunica media. This thin layer is the tunica media. This is the tunica media. And outside the tunica media, this very thick layer is the tunica adventitia, so which contains the vasa vasorum and nervi vasorum. So this is the tunica media. This is the tunica intima. And this is the tunica adventitia. We have also a Verhoeven staining of this section. So this very thin layer is the endothelium and subendothelial connective tissue, and this black squiggly line, it indicates the internal elastic lamina. And this is the tunica media, which is mainly composed of smooth muscle, circularly arranged smooth muscle. But in addition to the smooth muscle, we can also see elastic fibers. And this is the tunica adventitia, the outer layer. And in the tunica adventitia also there are elastic fibers. And between the tunica media and tunica adventitia, this line indicates the external elastic lamina. This continuous squiggly line, it indicates the external elastic lamina. Similarly, we can also see the vein. So this is the tunica intima with endothelium and a thick subendothelial connective tissue, which is pathological. Normally it's very thin. And this is the tunica media. But this very thick layer is the tunica adventitia. So in the tunica adventitia, we can also see elastic fibers. So you can use this slide to summarize the 4 types of vessels. So when we see elastic artery, it has thick tunica media, which is composed of circularly arranged smooth muscles and many elastic laminae that run in between the smooth muscles, but it has a thin adventitia, tunica adventitia. Similarly, muscular artery also, it contains a thick tunica media with circularly arranged smooth muscles and elastic fibers and external elastic lamina. And the adventitia is thin in muscular artery. And when you see vena cava, the large vein, the tunica media is very thin. It contains only few circularly arranged smooth muscles, but the adventitia is very thick. It contains longitudinally arranged smooth muscle and connective tissue. Similarly, in collecting vein also, it contains a thin tunica media with few circularly arranged smooth muscles and a very thick adventitia, which is mainly composed of connective tissue. Now let us see the smallest blood vessels, arterioles, venules, and capillary. So when we see the arterioles, the wall of an arteriole is composed of endothelium. Then we have the tunica media with 1 up to 3 layers of smooth muscle. And we have the adventitia or the connective tissue that blends with the surrounding structure. And one of the distinguishing features of arteriole is the diameter of the lumen. It's roughly equal to the width of the thickness. So we can see it here. This is an arteriole, and this one is also another arteriole from another section. So they are composed of endothelium, then 1 up to 3 layers of smooth muscle, and adventitia or the connective tissue that blends with the surrounding structure. But when we see the venule consists of endothelium, and outside the endothelium we'll have the adventitia or connective tissue. Sometimes we may see very few muscle fibers, but they are very irregular and they will not form a layer. And the lumen of a venule is irregular and collapsed. As you see, the venule has a large lumen and it's irregular and collapsed, but the arteriole has a well-demarcated rounded lumen, and the diameter of the lumen is almost equal to the width of the thickness. So this is a venule. It's composed of only the endothelium and connective tissue. And here, this very small vessel is the capillary. So capillary is only composed of endothelium. And the lumen only fits a red blood cell. The diameter of the lumen, it's 5 up to 10 micrometers, so it only fits 1 red blood cell. So this very small blood vessel is a capillary. So let's see mesentery. So this mesentery, it consists of different types of blood vessels. We can see muscular arteries, collecting veins, and we can also see arterioles, venules, and capillaries. So you can use the mesentery to identify different types of blood vessels. So this is an arteriole, which is composed of endothelium and 1 up to 3 layers of smooth muscle, and the adventitia blends with the surrounding structure. So this is one arteriole, and this one is a venule. So as you see, it contains only endothelium and connective tissue. So this is a venule. So this is also an arteriole. This one is another arteriole with 1 up to 3 layers of smooth muscle, endothelium, and connective tissue. This is a capillary. As you see, the wall is composed of only endothelium, and the lumen only feeds a red blood cell. It's 5 up to 10 micrometers. It only feeds one red blood cell. And now let's see a venule. Let's look for a venule. So this is a venule, and this is an arteriole. So the venule only contains endothelium and connective tissue. Here is also another venule. This is a venule, and this is an arteriole. This is also an arteriole. So look around and try to find arterioles and venules. The last type of vessel is lymphatic vessel. Here in the section of mesentery, there are lymph nodes. There are many lymph nodes. These darkly stained structures are lymph nodes. So lymph vessels are found adjacent to lymph nodes. So let's zoom in and see this lymph node. So these are lymphatic vessels. And the wall of lymphatic vessels, it only consists of endothelium. It's only composed of endothelium. And like venules, their lumen is irregular and collapsed. But one of the distinguishing features of lymphatic vessel from venule is we cannot see blood cells in lymphatic vessel. Either it's empty, or we can see this pinkish substance, which is lymph. But we cannot see red blood cells in lymphatic vessels. Sometimes we can see these floating nuclei. These are nuclei of white blood cells. So lymphatic vessels, their wall is only composed of endothelium. Their lumen is irregular and collapsed, and we cannot see red blood cells in the lumen. We may see these floating nuclei of white blood cells. That's all about today's lab session. We have covered these slides, studied these slides and practice identifying structures, use both H&E staining and Verhoef staining to identify different types of vessels. And good luck on your assignment, and I will see you next time.